So, good evening. So I'd like to thank members of the association for support of the laboratory and also for coming out this evening on such a freezing night. When I was recruited by Bruce and Jim in 1993, I was lucky enough to meet Jim in Cambridge, England, where I was working at the time. And he said, come to Cold Spring Harbor, come and work for 5 to 7 years, and then the world's your oyster. So now, 20 years later, here I still am, and that's really because this is just an extraordinary place to work. And every day I really enjoy coming to work, and it's fantastic. So I'd like to talk a little bit about some of the influential scientists who've come through the Meetings and Courses program here at Cold Spring Harbor, and then explore how our educational activities intersect with progress in genome science through our meetings, courses, and our publishing efforts. So the Meetings and Courses program hosts 25 to 30 conferences in this auditorium annually, mostly on a biennial basis, each attracting between 100 and 500 participants. We host around 30 laboratory and lecture courses on themes that span the life sciences that are taught in small classes in teaching labs around the campus and at Banbury. These programs are generally organized and taught by outside scientists who come from more than 50 countries annually to participate. However, as you've heard, these programs synergize closely with year-round research at the laboratory. The program relies heavily on NIH, NSF, and foundation funding, attracts around 9,000 scientists every year to Long Island, and makes up approximately 10% of the laboratory's budget. Because the scope of the laboratory's meeting and courses covers much of biology, we eagerly anticipate the announcements from Stockholm each fall. In 2012, we were pleased to see that both the medicine and chemistry Nobel Prizes were won by alumni of our program. Shinya Yamanaka was a student in the 1995 cell biology course taught by the then instructor David Spector, now head of research, while British scientist John Gurdon has been a frequent lecturer at our frog development course and a regular attendee at meetings and symposia. The chemistry prize went to 2 Cold Spring Harbor meetings alumni, including Brian Kabilka, who we can see here after giving the keynote address at one of our Cold Spring Harbor Asia meetings in Suzhou, China. In 2013, we actually went 3 for 3 with the medicine prize, with Randy Sheckman and Jim Rothman both having taken the yeast course many years ago, while Tom Sudoff was a founding organizer of the meeting. We started in 2004 that focused on synapses. These are the junctions between nerve cells in the brain. Many influential scientists have taken or lectured in our courses, or organized or attended meetings. For example, a total of 7 student alumni of our courses went on to win Nobel Prizes for, respectively, prions, the first ion channel structure, green fluorescent protein, the ribosome structure, stem cells, and most recently, vesicle trafficking. One can argue that the research training they received at Cold Spring Harbor propelled them on their path to scientific success. Further scientists were founding organizers of Cold Spring Harbor meetings that were started many years before the field and these individuals were actually awarded the Nobel Prize. Other influential scientists who have organized meetings here include the presidents of Howard Hughes Medical Institute, Memorial Sloan Kettering, and the Rockefeller University. So let me switch now to genome science education. The lab has had an enormous impact on genome science through its educational activities, including the sharing of new results and data at our main campus and Banbury meetings, and publishing these results in the journals produced by Cold Spring Harbor Press. Meetings also serve as a place where people can trade ideas and discuss results with their peers, and continue to forge new collaborations with people they meet at Cold Spring Harbor. The lab courses serve to disseminate concepts and technologies through hands-on training and through the laboratory manuals that the press produce, and which are famed for including protocols that actually work. So I want to cover a little of how education in genome science got started here at Cold Spring Harbor. This is very much tied to the history of the Human Genome Project, of which Jim Watson was the first director. The picture here shows the cover of an abstract book from the 1986 Cold Spring Harbor Symposium. These booklets contain the conference program and the one-page synopses of the work presented by scientists at the meeting. In contrast to the rather lonely idyll beside the harbor that the picture suggests, the meeting was actually extremely popular and crowded, and as evidenced by the gathering here on Blackford Lawn. Incidentally, you can see another future Nobel Prize winner, Kary Mullis, the inventor of the polymerase chain reaction, a major tool for the coming genome projects, here enjoying cocktails on Blackford Lawn. This was an enormously important and influential meeting in many ways, not least because a panel discussion on sequencing the human genome was held in the newly constructed Grace Auditorium, which we're all in. This was the first properly public discussion among scientists about the proposed project. And you can see here not only moderators Paul Berg at the podium, with Wally Gilbert standing off to one side, but also David Botstein and Eric Lander, who's now head of the Broad. The proposal sparked controversy among scientists over whether this was money well spent. It was, after all, biology's first big science project, similar to the moon landing, super collider, or space station, estimated at least then to cost $3 billion at the time. Wally Gilbert raised the stakes by defining this project as the holy grail of human genetics. Cold Spring Harbor and Jim Watson were at the very center of these discussions, and the lab took the initiative to start a new meeting series on genome science. So in May 1988, the first genome mapping and sequencing meeting was held. And following the success, plans were immediately made to hold the meeting as an annual series in May every year. The following year, an important private meeting of senior scientists was held at the Banbury Center under the leadership of Jan Witkowski. At this meeting, the National Institutes and the Department of Energy divided up responsibilities for funding the Human Genome Project. The first year of the Human Genome meeting featured a chromosome and genetic map on the abstract book cover, as you'll see here. But by the following year, the organizers were responding to Wally Gilbert's Holy Grail idea and depicting leaders in the field as knights of the round table at the court of King Jim. The three knights depicted here are from left to right, Maynard Olson, Charlie Cantor, and Rich Roberts, who were organizers of the first couple of meetings. The idea that future benefits of the project would lead to cures for genetic diseases are hinted at by the signpost on the far right to CF for cystic fibrosis and H.D. Huntington's disease. It's important to understand that the Human Genome Project was a consortium of scientists from around the world at centers throughout the US, but also in the UK, Germany, France, Japan, China, and Australia. This list shows some of the major genome centers at which work was ongoing, including Cold Spring Harbor Laboratory, as Dick has pointed out. The annual Cold Spring Harbor meeting quickly became the gathering place for these groups to congregate and trade results and ideas. The proximity to New York airports meant easy access from pretty much anywhere in the world, and once here, the environment was a boon to fruitful exchange and discussions. By the late 1990s, the Public Human Genome Project was making rapid progress, but just before the start of the 1998 meeting, Craig Venter announced at Cold Spring Harbor an ambitious plan to complete the sequencing of the human genome in just 3 years, and fully 4 years ahead of the international consortium. Leadership of the public effort, NHGRI and UK's Wellcome Trust, immediately issued a press release, which was actually faxed from my office in this building, to accelerate their efforts in a new 5-year plan and complete a rough draft by 2000 and a complete sequence by 2003. By 2000, the two groups were no longer at war, and Francis Collins was able to state that the only race we're interested in today is the human race. Because of the disparities in science funding, the lion's share of the project was being done by a small number of large genome centers. In this pie chart, the contributions to the final human genome sequence pretty much reflect the attendance at the Cold Spring Harbor meeting, where scientists from Wellcome Trust Sanger Institute, WashU, Baylor College, the Joint Genome Institute, and MIT's Whitehead dominated proceedings. In 2003, because of the anticipated completion of the Human Genome Project, Bruce opted to hold the Genome Meeting as our annual flagship symposium. Here we can see Francis Collins delivering the Dorcas Cummings address to a packed house. The main announcement, not unsurprisingly, was made at the White House and made the covers of Nature and Science. But Cold Spring Harbor was definitely at the very epicenter of the celebrations that year. Of course, the Human Genome Project was by now essentially complete, though plenty of gaps still remained in the sequence. The bulk of the work now turned to analysis and annotation of the human genome sequence, that is, figuring out how the sequence was regulated and how it was read out. And of course, there were thousands of other species, animals, plants, and bacteria to sequence, reflected here in the 2005 abstract book cover that had now moved on from the Holy Grail theme of the earlier covers. Each year at the Genome Meeting, a panel discussion on the ethical, legal, and social implications in genomic research is convened. This panel debate changes in scope, for example, the patenting of genes in 1998, the history of the American eugenics movement in 2001, direct-to-consumer testing marketing in 2008, and last year's debate on non-invasive prenatal testing, an issue which currently is very much in the news. If we look back over the 25-odd years of Cold Spring Harbor Genome Meetings, we see that the meeting has grown to reach capacity by the late 1990s. Through the use of additional seating, tents, and closed-circuit television, we've been able to squeeze more than 500 participants into the meeting, despite the fact that this auditorium actually only seats 360 people. At the same time, the amount of work presented at the meeting in the form of talks and posters has generally increased along with the rising attendance. Another way to visualize this, and I'd like to thank Jan, my colleague, Jan Witkowski, for giving me these slides, is to visualize this, is to look at the increasing width of the abstract books shown in this complete collection of books. And in fact, if we compare the abstract books from the first year and last year, 2013, we can see that the meeting now attracts almost 3 times as many participants, almost 4 times as many abstracts, and generates almost 8 times as much in revenues. Many of these abstracts are presented as posters, similar to what one sees in a high school science fair, though the content is obviously much more sophisticated. The planned Nichols Biondi poster pavilion will greatly enhance the poster environment for presenters and participants alike. By the early 2000s, the Genome Meeting was regularly oversubscribed, and so we began exploring online broadcasting to allow others to virtually attend through our Leading Strand portal, which allows viewers to subscribe and watch the meeting from their computers anywhere in the world. And this has really taken off in recent years, with more than 120 site licenses sold to labs around the world last year. Another recent phenomenon is the emergence of social networking associated with the meeting. This is best exemplified by the use of Twitter, and this graphic shows the volume of tweets as a function of the meeting session. We allow the community to self-police this activity, and each speaker states whether their talk is tweetable or not. As you can see, the volume of tweets basically mirrors the oral sessions. And this is a word cloud showing that the bulk of the tweets, though certainly not all, are about the science being presented. Enthusiastic tweeters, including some of the current organizers of the meeting, really believe this adds value to the scientific discussions, and I'm beginning to agree with them. Part of building the community of genome science is about having some fun with the science. The Genome Meeting has really exemplified this. So including you and Bernie's Gene Sweep lottery game, in which meeting participants had to predict the number of genes in the genome. The price of the bet increased as the Human Genome Project neared completion, and the lucky winners were announced at the 2003 Genome Symposium. In 2005, a monopoly game was devised based on the HapMap project, which was measuring the extent of human genetic variation. And in 2008, we devised the genome crossword with esoteric clues and about science and scientists. Of course, much of the science presented at the meeting is reported in newspapers and scientific journals, but it is these human interest stories about scientists having fun that have often generated the most public interest. So finally, after the banquet each year, and until very recently, Francis Collins' band has provided musical entertainment. In fact, the genome guitar that hangs in Blackford Bar was played by Francis at the 2003 symposium. Since becoming director of NIH, I'm afraid he has stopped playing at the meeting, but I would like to play a short video clip of him talking about the Genome Meeting.
Cold Spring Harbor has been, for me, and I think for anybody involved in the core of what genomics is all about. This is the center of where you gather to tell your colleagues about what has happened that's new and exciting. I've come to the genomics meetings at Cold Spring Harbor for, gosh, ever since they've been genomics meetings at Cold Spring Harbor. I don't think I've missed one. And they are clearly the moment during any given year where the most important, exciting presentations and conversations occur, because this is seen as the place where you want to be if you're really going to be talking about what's happening and what will happen. So many other disciplines have their meeting. For genomics, it's Cold Spring Harbor. It has been from the beginning, and nothing else has ever really made a serious competition for that niche. And I hope that continues. I think this is the kind of environment, and it's an international one, especially for this field, where people do come with a sense that there's going to be openness, that there's going to be great comfort with people standing up and challenging a particular perspective, that young people are welcome, that old codgers are welcome as well, and that they can interact with each other and there's no barriers to that. This is the sort of idyllic way that science is supposed to happen, and Cold Spring Harbor provides that opportunity, and I think it's made a big difference. If we hadn't had this venue, I'm not sure we would have negotiated all of the many complications. I mean, I can look back personally at the times where really difficult things had to be decided, and many of them got decided in conversations at the bar or out in the grass or sitting by the harbor right here.
So genome research has grown very rapidly in the last 2 decades. And so from the mid-1990s onward, the lab has hosted a growing number of meetings on subtopics and specialized disciplines around genome science. In fact, each year we now host 5 to 6 of these meetings annually and attract around 2,000 scientific visitors. Most of these meetings occur on a 2-year cycle. In 2014, we'll be starting a new meeting on big data, a major new theme in biological sciences, organized by Cold Spring Harbor scientist Michael Schatz and others, including the head of Amazon Web Services. In parallel with the lab's genome meetings, we embarked in the early 1990s on a series of hands-on short courses in genomics. These courses last between 1 and 3 weeks of highly intensive instruction and feature small classes of 15 to 20 students, with plenty of faculty participation from the best universities around the country and world. Because many of these courses have a significant wet lab or computational lab component, they're necessarily expensive to run, and so we rely heavily on federal grants from NIH and NSF and from foundations such as Howard Hughes. Each course has its own niche and character and culture, and in fact, Dick McCombie was instrumental in starting the Advanced Genome Sequence Analysis course, and I think it was 1995 that it started, which now is in the 20th year and will be offered again this fall. As Dick emphasized, the methods, time, and cost of sequencing has changed so dramatically that the first and most recent courses doubtless share little or no common curriculum. So what is unique about these courses, and all the courses, is that the trainees are senior graduate students, postdocs, and even junior or senior faculty, and the methods and ideas being taught are at a very advanced level and are updated every year. Here we can see classes from several years of the DNA sequencing course that Dick taught and still teaches. The courses are often 3- or 4-fold oversubscribed, and so the instructors who teach can select outstanding students. Because everyone stays on campus for the course, there's ample opportunity for informal exchange and discussions into the small hours, and many students and faculty go on to collaborate with each other. If you ask a student at the end of one of our courses what they most want to do, the answer is usually sleep. But ask them again several years later, and they will likely cite this experience as a highlight of their scientific career so far. There are a couple of quotes here, which I won't read, but regarding the impact of the DNA sequencing course. The course is also synergized with year-round research at the lab. Cold Spring Harbor scientists can sit in on any of the lectures or even fully enroll. And the program introduces us, that is the research community here, to new ideas, techniques, and to people. Cold Spring Harbor researchers can also connect with the companies who provide expensive equipment and reagents to the courses. So I'll close by emphasizing that our colleagues at Cold Spring Harbor Press also publish manuals and scientific journals in genome science, perhaps best epitomized by the peer-reviewed monthly journal Genome Research. The journal publishes about 150 papers annually, boasts an elite editorial board including the head of the National Human Genome Research Institute, and is ranked second amongst all genetic journals in the world. The journal is also keeping pace with the open access movement, and more than 30% of the articles are freely available without restriction. So in closing, I hope we have convinced you that the lab not only does groundbreaking and world-beating genome research, but that through our educational programs, we also deservedly merit being called the Crossroads of genome science. Thanks.